I'm going to start the recording, and I'll go ahead and introduce you all. So welcome everybody. Welcome to the workshop. My name is Jen Much, and I'm part of the CASE Board of Directors, and I'm helping and moderating with us today for this session. So on behalf of everyone involved with this year's conference, we want to thank you for joining in today. And your presenter, Tim Herman, has put in, and his team here, have put in a great deal of work to prepare for a magnificent professional learning experience for you all. And just I want to tell you about a few housekeeping items. Please be respectful and open-minded in your comments. Please engage in the session to get the most out of it as possible. And at the end of the session, probably later on today, you'll get an opportunity to complete a survey. And this will do two things, which will provide valuable feedback to us and serve as a means of tracking your hours to provide you with a certificate of attendance. So without further ado, please help me in welcoming Tim Herman and his team.
Hello, everyone. I'm Heather Ryan. I'm going to jump in here before we turn it over to Tim. Just to let you know, so I'm going to be here with Tim, and I'll be helping him with models and doing cameras and things and audio. And so if you have any problems with that, with seeing or hearing, let us know. And Chris is going to be helping us monitor chat during the session. And so you go ahead and post your questions there, and we'll try to address them as we go. Some of you were able to pre-register a couple of weeks ago and receive free student modeling packs. Can you go ahead and raise your hand or somehow let us know if you've received any of those? Forgot my Zoom etiquette. You can raise your hand. Okay, so it's okay if you didn't. Chris is posting in chat a link for you to register to get a free student modeling pack. And Tim's going to be doing some activities along with that in the course of the session today. If you have one or have some bone nucleotides, go ahead and assemble the sequence that is on the screen. We created these student modeling packs at the beginning of the pandemic in order to give students a chance for some hands-on modeling either at home or school. And they are our individualized versions of our most popular classroom kits that are all designed for group work. So now I have the honor of introducing my father, Dr. Tim Herman. He earned his Bachelor of Science in Chemistry from the University of Nebraska, his PhD in Biochemistry from Oregon State University, and then pursued postdoctoral studies in molecular biology at Harvard Medical School. Tim then joined the biochemistry faculty at the Medical College of Wisconsin, where his research areas range from the synthesis of chemically cleavable biotin-labeled nucleotide analogs to the development of novel approaches to structure-based drug design. After almost 20 years of research, teaching graduate and medical students at the Medical College of Wisconsin, Tim moved to the Milwaukee School of Engineering, where he founded the Center for Biomolecular Modeling to develop instructional materials, provide professional development, and deliver outreach programs. Shortly thereafter, he founded 3D Molecular Designs to make models, kits, and materials available to teachers and students. He has spent the last 20 years developing molecular models and kits and communicating fascinating molecular stories to educators, students, and the public. His efforts now reach more than 1 million students a year around the world. And now we'll begin our program. Take it away, Tim.
Okay, thank you, Heather. There's something really weird about being introduced by your daughter, but I'll try to get over that. So welcome to the workshop. I congratulate all of you for making time in a Saturday at the beginning of a school year that I know must be pretty difficult for just about everyone. Making time to sit through this session. We're going to try to make it informative, and I hope you'll find it interesting. We're going to be talking about CRISPR. And you know, I used to say CRISPR was the hottest thing in biology. And maybe now it's the second hottest thing because COVID-19 has sort of taken over where CRISPR was a year or so ago. But CRISPR is still there. It still is revolutionizing the way we do biology. And we are going to start using that technology in the very near future to start editing the human genome. So the first question is, you know, should you be teaching CRISPR to high school students? I think the answer is yes. Not necessarily to freshmen in their first exposure to biology, But certainly in some of those upper levels, say AP biology or advanced biology courses, I think it's a really important topic to teach just because, as I said before, this has revolutionized the way research is done. And it's going to continue to be really useful as a gene editing tool into the future. So how do you teach CRISPR? It's complicated, all right? So I'll tell you right now, it's not something that you're gonna sit through this hour-long session and you're gonna know everything you need to know about CRISPR to teach it. Unfortunately, that's not going to happen. And in fact, if this next hour goes right, then I hope you have lots and lots of questions, additional questions that you want to ask and figure out about how does CRISPR really work. I'm going to be covering a lot of ground today. Is this first slide up?
Yes, it is.
Okay. So the first slide, there are some questions, and we're going to try to cover these basic topics today. So it's all about how does CRISPR Cas9 work? So this protein I have in front of me here, which you can't see, is a 3D printed model. Now they can see it.
Now they can see it.
I can't see myself. This is a physical model of the CRISPR Cas9 protein. So the protein that most people are talking about when they talk about CRISPR and genome editing is this protein right here, Cas9. So we're going to talk about how does this work? What does it do? How does it work? What makes it special? And then some sub-questions under that, going back to the other slide now maybe. We're going to first consider CRISPR as an adaptive immunity system in bacteria. So this is where CRISPR evolved. CRISPR evolved in bacteria. Cas9 is only one of many, many, many CRISPR systems, each one evolving in a different bacteria over the years. And we'll talk a little bit about how it works in bacteria to protect them from bacteriophages. All right, and then after we've done that, that's so-called CRISPR biology. This is how CRISPR works out there in the wild. We have then taken this Cas9 enzyme out of that bacterial system, and we are now beginning to use it to make specific cuts in DNA, and even in the human genome. And I'm going to give the punchline all the way right now and tell you that the thing that makes CRISPR Cas9 so special is that it can seek out and find and cut a absolutely unique sequence in the human genome. And that may not mean much to you or to your students when you first hear it, but in fact, that is a very, very, very remarkable thing. This is a tool that molecular biologists have been waiting for, not even realizing it was possible, but in the last 10 years or so, as it became known that such an enzyme existed, people are just going crazy and figuring out how they can use this new technology in all sorts of very, very clever ways. So we'll be going over two kits then, two schematic-based kits that 3D Molecular Designs has developed to help you, the teacher, introduce your students to how the CRISPR system works and how the Cas9 protein makes this sequence-specific cut. And then after that, we're going to get to this third question that's listed here, and that is what happens after the cut? And this is the point at which people are often disappointed with CRISPR, because we talk about it, going into all these details about how it makes the cut. And then, but wait a minute, is that genome editing? That's just making a cut. What we really want to do as we edit the genome is either to knock out a gene, to inactivate it, or more importantly, we want to fix it, maybe by inserting a new gene into that cut site. So the fact of the matter is, CRISPR is not very good at what happens after the cut because it's not really involved in what happens after the cut. So we'll talk a little bit about your endogenous repair systems that spring into action and try to repair that damage that you've done to your DNA. But then we will spend the last, probably 10 minutes talking about how this Cas9 enzyme is being engineered now to make it a better genome editor. And this is where really, really clever molecular biologists come into play, young molecular biologists who have come up with 2 new ways to use CRISPR. One is called base editing. It doesn't require cutting the DNA. And the other is prime editing, which is really the application that you're going to find CRISPR to be useful in the future in editing our genome. So that's what we're going to try to do today without causing too many questions. There's this great quote, you know, we all seek complete understanding, but we're grateful for a momentary stay against confusion. So we hope to give you an inkling as to what CRISPR is all about today. At the same time, tweak your interest a little bit in finding out more about how this system works and how it's being applied. All right, can we have the next slide?
Yeah.
We're going to start. Oh, can I do this?
So you wanted to be able to zoom in here.
Yeah.
Correct?
Yeah.
Go ahead.
Yeah, all right. So we put together, Mark in our group has put together a series of 3 graphics that kind of go over what we call CRISPR biology. And that is, how does the CRISPR system function as an adaptive immune system in bacteria? And this is just the first panel of those 3 graphics. And I'm going to start with this and to sort of to explain an overall, this is how it works. And then we're going to go to a foam kit and show you how you can have your kids actually model this process, this adaptive immunity kit with the foam kit. So if you are looking at the graphic, then this is a bacterial cell. Here's the membrane. Here's a bacteriophage up here, a purple one. And you see that it's in the process of infecting this bacteria. And what that means is it is injecting its purple genome into this bacteria. And if left unchecked, this bacteriophage will often take over this cell and simply turn it into a factory that makes more and more bacteriophage. And ultimately the bacteria lyse, they break open and release thousands of identical copies of this purple bacteria. This bacteriophage. So the bacteria have been fighting this battle against these bacteriophage for millions and millions of years. So they have evolved a CRISPR system to protect themselves from these bacteria. So just very, very quickly, here's how it works. This system is actually a small section of the bacterial genome where you find some very interesting genes clustered together. And we refer to this as the CRISPR locus. And here's a blow-up then of the kinds of genes you find in a CRISPR locus. And I want you to look first at these black sort of diamond-shaped things here. These are short palindromic repeats. Now let's find some place where CRISPR is spelled out. Can you see the CRISPR down here? You see this SPR, short palindromic repeats. CRISPR is a great acronym that you either hate or you love. You hate it before you understand what it really stands for. The SPR letters stand for these black, short palindromic repeats. This was the first thing that people noticed when they first discovered CRISPR. Without ever knowing what it did, they simply noticed that there were these short palindromic repeats that were clustered together. That's where the C comes from, clustered together in the genome, and they were regularly interspersed with these other sequences, a blue sequence here, a tan-colored sequence here. So going back to CRISPR, they're clustered, regularly interspersed, short palindromic repeats. So that just describes this part of the CRISPR locus, a region in the bacterial genome where you have all of these genes which are going to work together in a way to protect the bacteria from this bacteriophage. And to make a very long story short and oversimplified before we go to the physical model, these genes are going to send out what's called an acquisition complex, a multi-subunit complex of proteins that's going to cut a small piece out of this genome, this purple genome, and it's going to bring that small purple piece. I know I'm giving it away. I'm giving you the answer right now. They're going to bring that small piece of bacteriophage DNA back, and they're going to insert it right into this CRISPR locus. And from that moment on then, in a way that will be revealed a little bit later in this session, it's now going to be able to deploy a CRISPR Cas9 equipped with a piece of RNA, which will allow the CRISPR Cas9 to target, to find and cut up this viral genome before it ever has a chance to complete the infection process. So that basically is CRISPR. That's how it works. No antibodies are involved. In our adaptive immunity system, we use proteins called antibodies. Bacteria use CRISPR, which is a RNA-guided endonuclease. It's a nuclease that's going to cut the bacteriophage DNA before it ever has a chance to establish an infection. Okay.
I'm asking you to move on because we're behind schedule
We're
already.
going slow. I tend to go into too much detail. All right, so can we have the overhead camera on here? So these are the major components of what we call the adaptive immunity kit. Up here we have 3 different bacteriophage, purple, blue, and tan. This is the cell membrane. And this purple bacteriophage has bound, and it's now beginning to insert its purple genome into this cell. Here is a representation of that CRISPR locus that we looked at in the graphic before. And I told you that we're able to reach out. There it is. I couldn't find my small piece of purple DNA. Now, scale is much different here, right? But just a short segment, about 32 nucleotides long, 32 base pairs long. Somehow magically at this point, I haven't told you how this happens, but there's a complex of proteins that go out and they just cut little 32 base pair segments of this purple genome out, and they bring it back magically. They have the ability to bring it back, and they're going to insert it into this bacterial locus just upstream from this last short palindromic repeat. They're going to put it in here, and they're going to put it in here. And from that moment on, this bacteria is going to be immune to that purple bacteriophage. But now, look at this for just a minute. And here's where you and your students are going to have to think critically. This is a very easy way to do it. These purple and blue and tan sequences actually turn out to be bacteriophage sequences. And it's like an immunization record for this bacteria. The most recently experienced bacteriophage is always integrated into this genome right here at the so-called left-hand end of this cluster of short palindromic repeats. So CRISPR is named for the short palindromic repeats, and these colored sequences in between were given the name spacers, as though they weren't really very important. But in fact, these spacers are the most important thing, because these represent sequences that identify a purple bacteriophage, a blue bacteriophage, or a tan colored bacteriophage. All right, so we've simply brought this purple sequence back, we put it in, but what's wrong? What's wrong is, how am I going to put in the next bacteriophage DNA? When we brought this one back and successfully inserted it, we were looking for this red leader sequence and a short palindromic repeat. And that's what the acquisition complex recognized in order to know where to insert this. But I just pulled this apart and inserted it. And now we don't have this short palindromic repeat up here. so we can put the next one in. Told you I was going to generate more questions than answers. So this is what really happens. The complex comes back carrying the purple DNA. It finds a place it wants to put it in. It cuts this phosphate ester bond here. And it cuts this one here. And then this short palindromic repeat. I did that wrong. It cuts it here and here. The short palindromic repeat, which is just held together by a few base pairs, that comes apart. And this is what you have. And this is where you put the purple spacer. You form a phosphate ester bond linking the 3' end of this strand, the 3' end of this strand, and then you've got a gap. And in the gap, then you have a 3' hydroxyl group, which then is extended nucleotide by nucleotide by nucleotide by nucleotide. About 29 nucleotides in a row get synthesized to fill in that gap. Same thing is happening over here. So now you see we have successfully inserted the purple sequence, and we've also duplicated the short palindromic repeat. So we're ready to do that again with the next virus that comes along. All right, great. How does this explain adaptive immunity? We now go on to the next phase here, which we then express this locus. We express all these genes to eventually come up with this Cas9 endonuclease that I showed you here. All right. Let's do it with these models first. We're first going to, an RNA polymerase is going to come along. It's going to express this orange gene. This is called a tracer gene. And we're going to make lots of pieces of this orange tracer RNA. This is not messenger RNA. This is going to be a structural RNA that's going to become part of that Cas9 protein. Another RNA polymerase is going to come along, and it's going to express the white gene, which is the gene that encodes the Cas9 protein. This is a messenger RNA. This messenger RNA for Cas9 is going to be translated by a ribosome into a Cas9 protein, which is represented by this white oval. And the ribosome is going to translate this many, many times. So you're going to make lots of Cas9 proteins. But these are currently non-functional because these now have to be equipped with an RNA. This is an RNA-guided endonuclease. Where does the RNA come from? Well, we've got some tracer RNA here, and now we've got the business end here, these spacer sequences that represent bacteriophages. So we're going to express all of this as one continuous RNA. Again, this is not a structural RNA. This is not a messenger RNA. This is going to be a structural RNA. This is a detail you don't need to worry your students about, but you make it as one long piece, and then you process it down into these shorter pieces like this. And now you see we have a piece of RNA that has the purple bacteriophage sequence plus a little bit of the black spacer, the short palindromic repeat. And then you've got the same thing here with the blue viral sequence, here a tan viral sequence. All right, things are starting to get a little complicated here because the 3' end of the tracer base pairs with the 5' end of the short palindromic repeat to make a dual RNA, which now binds in a very specific way to the Cas9 protein. And we now have a dual RNA-guided Cas9 endonuclease. This protein is now ready to go. This is the protein that is going to be able to search out incoming purple viruses and destroy it. At the same time, you can make and equip this Cas9 protein with a blue sequence. So it can now be on the lookout for the blue virus. And finally here, the tan colored virus is also deployed. The Cas9 is, you now made a Cas9 that can search out and destroy all 3 kinds of viruses. So this is the adaptive immunity system. And you remember these viruses that you've encountered in the past. And just like an antibody protein, you now have something that is on guard to protect yourself from those. That's CRISPR biology. We talk about Cas9, but it is literally one of only, dare I say, 1,000, certainly 100. There are hundreds of different Cas9 systems that have evolved in hundreds to thousands of individual bacteria over the ages that they've been fighting this battle against bacteriophages. Cas9 from Streptomyces pyrogenes. If you want to impress your kids, practice saying that. This is the prototypical CRISPR system because there's something very special about Cas9 as opposed to Cas12 or Cas13A or Cas many, many, many numbers. So Cas9 is what people will talk about, but know that it's only one sort of prototypical member of a large family of proteins that bacteria have evolved to protect themselves from bacterial viruses.
I'm going to jump in here for just a second. So I haven't noticed any questions in chat. We are about to move on to the next part. There any questions or confusions with this initial models before we go ahead and take them away? Okay, so I think that we are good and that you should go directly to your nucleotides.
Directly to my nucleotides.
Yes.
It's okay. All right. Heather manages me. She keeps me on schedule here. I think she's telling me I'm behind time. All right, you got, we're now going to talk about this enzyme, Cas9. What makes it special? And you're not going to just say, hey, students, today we're going to talk about Cas9. You're going to have to connect it to something that you've already taught them. This is why you can't teach this to freshmen, I don't believe. The best way to teach this is to kids who've already heard about restriction enzymes. You know, in the 70s, we discovered these restriction enzymes, which were able to bind to DNA and make sequence-specific cuts in the DNA. And coming out of that in the 70s into the 80s, this led to the recombinant DNA era because we suddenly could begin cutting DNA up into small fragments and we could clone them, we could sequence them. So this was the beginning of molecular biology. These wonderful proteins that we found in bacteria called restriction enzymes. And here's a model of a restriction enzyme bound to a very short sequence of double-stranded DNA. I know you can't see this very well, but I just want to make the point that these restriction enzymes recognize very short sequences of double-stranded DNA. Some of them recognize only 4 base pairs. This one recognizes 6 consecutive base pairs of double-stranded DNA. And they make cuts. All right, so we're going to do a short little activity here with your foam nucleotides. If you pre-ordered this, and if you had this, you might have made this sequence. So these are color-coded nucleotides: green guanines, blue cytosines, red adenines, yellow thymines. So it's a sequence. And you know, you and kids look at this and it's a sequence. So what? Notice that there's a 3' end here and a 3' end here. So the 2 strands are anti-parallel. That becomes very, very important in molecular biology. And I will be happy to argue with you in person at a summer course about the importance of teaching 5', 3'. If you're a serious teacher of biology and have serious students of biology in your classroom, you have to deal with this issue. All right, but let me point out something now. I'm going to take this sequence magically apart. I'm going to remove these 4 nucleotides and these 4 nucleotides, or these 6 nucleotides, so we can focus on these 6 I'm going to point out this sequence is GAATTC. Read the other way, it's GAATTC. This is a palindrome. This is the kind of sequence that these little restriction enzymes that microbiology provided to us. This is the kind of short sequence that a restriction enzyme can recognize and cut. Okay? But I want you to think about this. How frequently would you expect to find the sequence GAATTC in the human genome? Now that's a tough question to ask a group of students. How often would you expect to find these 6 nucleotides, GAATTC, in the human genome?
Go
And
ahead and post it in chat.
yeah, if you have the answer, post it in chat. But for the rest of us who are a little slower, we're going to reason through this. What's the chance that if you can download the human genome now, right? And you'll start looking at the sequence. And what are the chances that the very first letter in that big file is a G? Well, that'll be 1 out of 4, because it could either be a G, it could be an A, it could be, so it's 1 out of 4. What's the chance that if you have a G there, the next letter will be an A? That's also 1 out of 4. So what's the chance that the first 2 letters would be GA? That's 4 times 4, which is 16. So you have a 1 in 16 chance of finding a sequence that starts GA. And then GAA would be 4 times 4 times 4. 16 times 4 is 64. So you'd expect to only find GAA every 64 nucleotides in the human genome. And you keep multiplying by 4 6 times, you get a number that's a little bigger than 4,000. So if you had a random sequence, you would expect to find this 6-base sequence or any 6-base sequence once every 4,000 nucleotides. All right, now think about this. How big is the human genome? And that's something that every biology teacher just has on the tip of their tongue now, because we sequenced that genome back in, what, 2003? So we know how big the human genome is. It's 3.2 billion base pairs. That's 3.2 times 10 to the 9th. Now this restriction site, if it occurs every 4,000 base pairs, that's 4 times 10 to the 3. If you divide 3 times 10 to the 9th by 4 times 10 to the 3, you're going to get 800,000 ECO-R1 sites, restriction sites in the human genome. So if you took the human genome and you incubated it with a restriction enzyme, you would get a complete mess because it would be cutting all over the place. All right. This is what's special about CRISPR Cas9. This is like a restriction enzyme. It can make a sequence-specific cut in double-stranded DNA. But it can search out and find what we refer to as a statistically unique site in the 3.2 billion base pair human genome. Okay? So the question you ask your students then is, how long is a statistically unique sequence in the human genome? How many nucleotides would make up a sequence that you would expect statistically to only find once every 3.2 billion base pairs of random DNA? So the answer is simply 4 times 4 times 4 times 4 times 4 times 4. You keep multiplying by 4 until you reach a number that is equal to or larger than 3.2 billion. And the reason you made a 16 base pair sequence with your nucleotides is that this represents a statistically unique sequence in the human genome. So I found that fascinating when I first sort of started thinking about this. On one hand, I think this is an amazingly short sequence to be found only once in 3.2 billion base pairs. But then the next question becomes, how is Cas9 able to recognize something that's this long? You know, if there are 10 base pairs per turn, this is like 1.5 turns of double-stranded Watson-Crick DNA. So how is this Cas9 going to sort of wrap itself around, 1.5 turns of a helix? Over here, this is just a protein DNA interaction. This restriction enzyme binding to DNA. The amino acid side chains on the restriction enzyme are interacting with functional groups on the bases. So it's a direct protein DNA interaction. I'm giving away the punchline here.
I was wondering if you wanted to go click through those slides real quick, just so that people have a reference of what you've talked about.
Okay.
If you do it quickly, you'll be back on top.
All right. Over here. Yeah. So here are a few slides that just go through the nucleotide exercise we just went through. And this PowerPoint will be available following this, so you can go back over this. So here's the EcoR1 restriction site, GAATTC. Here's where it cuts. And we arrived at this number, 4,096 nucleotides is how frequently you'd expect to find this in random sequence. How did we get that number? Well, let's assume that number is right. If that number is right, and the human genome is 3.2 billion, then you divide those two and you get 781,250 fragments. So you'd make a mess of the human genome with EcoR1. In other words, a mess. All right, and then here's a question. CRISPR Cas9 cuts the human genome only once. How many nucleotides? The answer is, you have to guess. You don't have to guess, you calculate. And when you do the calculation, you find out that the answer is 16. And here's how you did the math. 4 times 4 times 4 times 4 times 4 gives 4.3 times 10 to the 9th, which is greater than 3.2 times 10 to the 9th, or a 16 nucleotide sequence is statistically unique. That's what Cas9 can do. Now the question is, how does it do it? And while Heather is giving me the next kit that we're going to use to look at this, here's a slide. I hope you're seeing this slide that shows this Cas9 protein. And the first thing I would point out here, the Cas9 protein is 1,368 amino acids long. Now, this is very important and at the same time, not important at all. It's not important at all that your kids know that Cas9 is 1,368 amino acids long. But it is very important that they know that it's large. Compared to proteins, this is a large protein. And it is a large multi-domain endonuclease. So the protein is modeled here just as a surface model, and you see all these knobs. Each one of these knobs could be a perfectly well-behaved individual protein. And in many of these CRISPR systems, they're exactly that. This Cas protein assembles from individual proteins into a multi-subunit complex that functions like Cas9. But Cas9 has evolved to be one single continuous polypeptide, which folds up as one protein, but it folds up into these different protein domains. And the problem with a model, a physical model,
Getting a little deep here now.
getting a little deep. That protein, this is hard plastic, right? This is a snapshot of the structure of Cas9. But what it doesn't convey is the conformational flexibility that this protein undergoes. So this is its structure only at one moment in time. Okay, and what kids have to develop a real appreciation for is how the shape of this protein may change as it searches for a bacteriophage DNA and then cuts the DNA. All right, if we can go to the overhead camera, then we're going to talk about how this Cas9 actually works.
Zoom this out for you.
Yeah. Okay,
Okay.
and I'm going to take this apart for just a second.
Slide this.
So are we sort of on time here?
I think you're okay.
All right,
We have
so we're going to take all this
about
apart.
a half an hour left.
Yeah, we're going to model.
Oh, okay.
We're going to model how Cas9 works in. And you see here, we have a placemat in which we have this white Cas9 protein, a schematic two-dimensional model of CRISPR Cas9. All right, and we're just going to set that aside right now because we know that we have to program this Cas9 protein with a dual guide RNA. Can you hand me the purple guy? Remember this is what we have to build now. We have to add some tracer RNA and some purple spacer with a little bit of the short palindromic repeat. This is the dual guide RNA that's going to equip that Cas protein to search out for a purple virus. So what your kids
Slide this forward a little bit more.
will have if you acquire this kit is they're going to have this orange RNA, and they're going to have 5 pieces, and that's just because the foam that we use to make this is only about this long. So we have to create this in 5 pieces, but then we show you how those 5 pieces go together to make one long piece of tracer
I have some
RNA.
nucleotides that are upside down there.
I knew there was something wrong. Okay, all right. So there's our tracer RNA. Now, tracer RNA, I trust you're not teaching that RNA is single-stranded and linear. That's old stuff. That's not true any longer. It never was true. It's just that we used to teach it that way. But this is going to fold up. It's going to form some base paired, some hairpins. There's an AU base pair. Here's an AU base pair. Here's a GC base pair. So it's going to start to fold both in 2 dimensions and then in 3 dimensions. And as it does that, it's going to bind to the Cas9 protein. So you start by putting the 3' end with the arrow. Right here on the placemat, it says 3' end UC. So here's UC, and it goes there. And then that helps you find this hairpin. See this base pairing here? So here's a hairpin. Here's ACA. So this fits right in here to the protein. So this tracer RNA is binding to the Cas9 protein. There's another hairpin here with some more sequence here to help your students find this. Okay, so that's the tracer RNA bound. That's one RNA. Here's another RNA. Here is that purple spacer sequence, continuous with some black short palindromic repeat. And here's the amazing thing. I told you that CRISPR is complicated. Okay, we have a poster somewhere behind me here that says that. The 5' end of the short palindromic repeat is complementary to the 3' end of the tracer. So you see these base pairs right here? So here's your dual RNA now. 1, 2 RNAs, which are now bound by the Cas9 protein in just such a way now that this purple RNA sequence, the guide RNA, this is going to interrogate a piece of double-stranded DNA to see if it represents DNA from the purple virus. And let's take one aside for just a second and look at the end out here. You know that Jennifer Doudna shared the Nobel Prize in chemistry last year. Not last year, 2 years ago now. We just have a new Nobel Prize this year. 2 years ago, Jennifer Doudna won the Nobel Prize, shared with Emmanuel Charpentier, because they converted this dual RNA into a single guide RNA by connecting the tracer to the CRISPR RNA with this tetraloop. So this is Jennifer Doudna's unique contribution to the field of CRISPR, is just simplifying this system so that what used to be a dual RNA guided endonuclease in nature to now a single guide RNA that's used by molecular biologists all over the world. All you have to do now is swap in different spacer sequences that you're using to target this to a particular sequence. All right, so here's a piece of sample double-stranded DNA that you want to cut if in fact it represents bacteriophage DNA. So I'm going to race ahead here, and I'm going to point out that right here on the placemat, it says there is a PAM recognition site. And I apologize, but PAM stands for Protospacer Adjacent Motif. And you should be shaking your head. What? I'll say it again, Protospacer Adjacent Motif. That doesn't help at all, does it? But I can tell you that with time, that is going to make all kinds of sense. And when this light bulb goes off in your head, you're going to smile. And I appreciate Jennifer for having her camera on, because it's always nice to at least see one person out there smiling or frowning or giving up. All right, so the Cas9 protein doesn't just start at one end of the DNA and look. It actually looks for what we call PAM sequences, which in the case of Cas9 is the sequence NGG. So whenever you have any nucleotide followed by GG in the 3' to 5' to 3' direction, only there will it bind. And then when it binds, this multi-domain protein is able to separate the two strands. And we're going to call the top strand here the target strand. And the bottom strand then is the non-target strand. And we're going to interrogate the top strand with our purple guide RNA. And if it is complementary, if there are Watson-Crick base pairs across this entire nucleotide sequence, then only then will this Cas9 protein adopt a shape in which it is enzymatically active as an endonuclease. So if this doesn't base pair like this, then this active site and this active site, which are each contributed by two different protein domains, you know, that multi-domain Cas9 protein. They're knobs of protein that are homologous in sequence to two separate endonucleases. And only if this base pairing matches does this become active, which cuts the phosphodiester bond on the target strand right here, and right here, it cuts the non-target strand right here. So we've cut both strands. We're going to disengage the guide RNA because we've now made the cut. All right, so this falls away. And what you're left with is the original double-stranded DNA, which in this case could be a bacteriophage DNA. And this, of course, will zip up right away because it's complementary. So here is a double-stranded DNA, which has been cut at a specific sequence, 3 nucleotides upstream from the PAM sequence, 1, 2, 3. And this sequence was now specific for the spacer DNA that you equip the Cas9 with. So that's how Cas9 works. It actually uses Watson-Crick base pairing between a guide RNA and the target strand of double-stranded DNA. And if those are complementary over that entire length, then these active sites work. And suddenly you have a tool that can then be used to make a sequence-specific cut in the human genome. I mean, this is the way it protects, the bacteria protects itself from bacteriophage. But we can now take this Cas9 enzyme and we can switch into it any nucleotide sequence we want. And we can target that endonuclease to any sequence we want in the human genome. So the use of restriction endonucleases in doing science is decreasing, because why do you want to buy, you might have to, in the past you had a freezer filled with 20 or 30 of your favorite restriction enzymes. In the future, you'll have a freezer with 1 tube of Cas9 and then a bunch of different spacers. Each one may have cost you, $10. So you have a whole new toolkit, much less expensive, much more economical, much faster, much easier to use than restriction enzymes. Okay, how are we doing on time?
You're doing great on time,
Good.
and I'm wondering if there are any questions.
Yeah, I've been doing a lot of talking, and if anyone wants to ask a question, raise your hand or put it in chat. I hope I'm not completely confusing people. Let me stop at this point and say one thing, and that is, I don't at all believe that if you didn't know anything about CRISPR and you came to this session, you're probably not going to go away from this session saying, yeah, I'm going to teach this tomorrow. But there is going to be a 5-session evening webinar series that will launch probably in March of next year, run through March and April. And we're going to go over everything I've talked about here in much greater depth in 5 evening sessions, at the end of which I hope you will see how you can incorporate this into one of your advanced biology classes. So we'll say more about that at the very end of this session. Are there any questions that have come up in chat, or if anyone wants to ask right now? Questions, comments?
I think you're doing great
I'm
and
explaining it
this.
so well that there are just no questions.
Actually, you have been wonderful. It's been wonderful. I did lose it just at the end. You, right after saying where you cut 3 nucleotides upstream from the PAN site, I guess I lost a little bit of where it maybe how it engages with the human genome. And is that just related to just the PAM site? Or
well, this Cas9 will work in the human genome just as it does in a bacterial system. That is, it will look for a PAM site. It will only be able to cut at a sequence that's upstream from a PAM site and complementary to the guide RNA. So it's not as though you can take Cas9 and make it cut anywhere at all. You first have to find a PAM site, an NGG, and then if you have equipped your Cas9 with a guide RNA that on the target strand is complementary to the target strand starting 3 nucleotides upstream, only then will it cut. All right, so it's not quite as versatile a tool as we might like it to be, but clever molecular biologists have gotten around that as well, because you can engineer the PAM site to change. There are different variants of Cas9 that recognize different PAM sites.
Or different Cas proteins,
Or
Cas12.
Yes, all the other Cas9-like proteins have different PAM sites. Now, there's something we've not talked about. We're not even going to try to get there right now. But let me just ask you, just ponder this question. If you're a bacteria and you have a CRISPR system, why doesn't your RNA-guided Cas9 cut your own genome up? Why doesn't it find this purple sequence in the CRISPR array in your bacterial genome and cut you up? I'm not going to answer
I'm
that
going to
question.
steal the camera away. Say that we're going to wait and maybe talk about that more at the end. We'll
Yeah.
let that percolate in the back of your mind.
We'll talk about that in the webinar
But
series.
how about we talk about what happens after the cut?
Yeah, this is my least favorite thing to discuss because are people looking at this?
Yes.
Yeah. What happens after the cut? The easiest answer is all hell breaks loose. And you have very little control over what happens after the cut. So all Cas9 does is make a cut. Now, I don't mean to minimize that. That's amazing that it makes one cut at a unique site in a human genome. But once it's done that, it doesn't automatically change the gene in the way that you might want to change it. But once again, our cells now, we're now switching to what we call CRISPR biotechnology. So how are we going to use CRISPR to edit our own genome? So we have been dealing with damage to our DNA over all of these years. And the only reason we're here to talk about it today is that we have evolved two basic redundant systems to repair our DNA. And one is called non-homologous end joining, which is a very rapid but error-prone repair system. And then there's this homology-directed repair that requires a homologous donor DNA. This is actually what happens, you all teach mitosis and meiosis, right? You teach crossing over during meiosis. So we have evolved a bunch of enzymes that repair those breaks and strand invasion, these DNA transactions that happen during meiosis. So these are, we have within all of our cells these enzymes which, given enough time, will repair the damage that sunlight does to our DNA, or in this case, a bacterial Cas9 enzyme has done. But if you want to knock out a gene, if you want to just inactivate a gene, then CRISPR Cas9 is a pretty good way to do that, because you cut the gene. As you try to repair it with this non-homologous end joining, you introduce insertions or deletions, which very quickly will inactivate the gene. So if you're just looking to knock out a gene, then CRISPR is a good way to go. But if you want to repair a gene, if you want to insert a good copy of the gene into a bad gene, then you have to provide this homologous donor DNA. And then you have to hope that this homology-directed repair system gets to that cut site before the non-homologous system gets there. So while there's no doubt but what CRISPR Cas9 can be used to insert new genes into these cut sites, it's not happening at a very efficient rate right now, not at a rate that would support clinical use of CRISPR in genome editing. There is, however, an answer to that problem, and we'll come to that very soon. The point is, neither one of these outcomes warrants the use of the word editing. An editor is someone who makes very precise corrections to text, to letters, based on some very stringent rules of grammar. And this genome editing, CRISPR, as we've described it so far, is more like DNA abuse than it is DNA editing. Because once you make the cut, you really don't know what's going to happen at any one of those cut sites. All right, so that's the reality of where we are. But I will go right ahead and say clever molecular biologists like a guy named David Liu at Harvard, they've come up with not one, but two clever modifications to Cas9, which get around this DNA abuse and really warrant the use of the word editing when it comes to genome editing. All right, is that where we're ready to go now?
Yes, so we have 10 minutes to talk about editing and answer any questions before we do our.
Okay. Can I advance the slide? Yeah. All right, David Liu, minding his metaphors. Okay, if you don't like editing as a metaphor, David has developed something called base editing. No cutting necessary. Almost no cutting necessary. We may not get to the almost. But what is base editing? All right, so here's something that your students are going to love. This is the chemical structure of cytosine.
You're gonna love this.
Well, here's a reason. Here's a reason that your kids might want to know this. Here's cytosine, and here's uracil. Okay, you see this amino group here in cytosine? If you remove this amino group, you get uracil. Now, there is an enzyme that has evolved called cytidine deaminase. It's an enzyme that enzymatically removes this amino group to convert cytosine to uracil. So what David has shown is that without cutting the DNA, you can take this Cas9 protein with its guide RNA. You can mutate the two active sites.
Let's do this on the mat.
Do this on the mat. Okay, yeah, let's do this on the mat. So the overhead camera is coming back on. Here's our Cas9. So here's our DNA, okay, bound at the PAM site here. Two strands are separated. And now we're not going to take time to base pair this, but you know, the top strand is busy being interrogated by the guide RNA. But I want you to look at this bottom strand now, the non-target strand. And I want you to look for just a minute at the physical model. The physical model, I haven't really shown you this. I'm going to take this domain away. And I don't know, this is really busy here. Is there a white? Can I have that white? Yeah. Yeah.
Blow out when whites, but.
Yeah. So you see the DNA is a dark blue and a light blue strand, double-stranded DNA. And then right here, the two strands separate. Right? So the PAM site is down here. Two strands separate. The top target strand is interacting with the guide RNA up here.
Pointers over there.
Yeah. So this is deep inside the protein, especially if we add back this protein domain. Okay, but look at the non-target strand. The non-target strand is just sitting in this trough, pretty accessible to the outside. All right, so guess what David Liu has done? David Liu has created a fusion protein between the Cas9 protein and that cytidine deaminase enzyme. And all that means is that the C terminal end of the cytidine deaminase was continuous with the N-terminal end of the Cas9 protein. So in a base editing version of Cas9, you have another protein domain here. And that's represented on our schematic model here by this. So here is cytidine deaminase. And there's a small short tether here of about 6 to 8 amino acids. And that allows this cytidine deaminase to swing around here. And if it finds a cytidine, it's going to deaminate that cytidine. And it's going to change it to a U. Now, I don't quite have a U handy yet, so I'm just going to turn this cytidine over just to indicate that we've now changed a C to a U. Okay, and now this is going to fall away. Okay, so now we have edited a very specific base in a very specific gene at one base without ever cutting a phosphodiester bond. So all hell has not broken loose.
So what you didn't do on this model is take off your endonucleases.
Oh, yeah, that's what I was going to do. Before you did this, you inactivated these 2 active sites. So no cutting occurred once this spacer found the right gene that you wanted to edit. But now life is complicated because let's think a little deeper about this. As this DNA comes back together again, this is a lot to do to you guys. Right here, we used to have a GC base pair, but now we've edited this C to a U. So we now have a mismatch right here. The rest of this DNA, we've not damaged in any way. We've simply edited it very precisely, a C to a U. But guess what? When you have a mismatch like this, we have evolved what is called a mismatch DNA repair system that will recognize this inconsistency in double-stranded DNA, and it's going to fix it. Now, the question is, which one is right? Which one is it going to change? It could change this G to match this U, or it could change this U back to a C, so it would. And it's even worse than that. Because this U shouldn't be in DNA. And in fact, when we made the very important decision to switch from the RNA world to the DNA world, we realized that we'd have to change all of our U's to T's. And we did that. And from that moment on, we evolved an enzyme called uracil N-glycosidase, which will very quickly remove any U that it finds in DNA. So David Liu came up with this very clever way to convert C's to U's. But now he's fighting years and years of evolutionary biology, which has evolved an enzyme that will take that U away and replace it, change it back to a C. So what do you do at this point? Do you throw up your arms and say, it's good try, but we're defeated? No. Is this on the next slide? Let me see.
Yeah, I have to share the screen again. Give me a second. And we have just about 2 minutes to wrap this up.
All right.
And we know we aren't going to finish this to make sense. We're just giving you an idea of where we can go from here.
If not complete understanding. All right, here's David Liu. Here's what he's done. Here's the cytidine deaminase, which he has fused to the Cas9 nickase. I'm just giving something away here. But you see these 2 proteins down here? And at the C-terminal end of Cas9, he's added 2 more proteins. He's added 2 copies of another protein called the uracil glycosidase inhibitor. So just as there is an enzyme that will take this out, there are also enzymes, proteins, which are inhibitors of this. So you localize to this region in the genome where you've made this base edit 2 copies of an enzyme which will inhibit any of the uracil glycosidase that comes along, trying to fix this. At the same time, you add back this active site,
Keep going.
which nicks just this strand. So I said almost no cutting, because that mismatch repair system uses this as the clue to tell it which of these two it should fix. So if you can keep the U in place, you can then trick the cell into modifying this G to match the U. So you end up with an AU, eventually an AT base pair. Clever, clever, clever. All right. Very, very complicated. But for some of your advanced students, they're going to really get a kick out of this if they can really see the cleverness of this. And if you liked this, base editing is not what we're going to use in the future, because there is an even more clever modification to Cas9 called prime editing. And you can, in fact, model prime editing with this kit. I'm convinced that you can. We haven't quite successfully done it to the point where we can show it to you yet, but this next winter in March and April in that webinar series, I promise you, we will model prime editing with this CRISPR Cas9 kit. And that's the technology we're going to use to actually insert new genes, because the homologous DNA that we want to insert is going to become part of this tracer RNA. So it's going to be right there in the same place where you want it when you're ready to insert a new piece of RNA or DNA in this case. So clever, clever things. Teaching CRISPR is not for the faint of heart, but it's going to require some head scratching and thinking. It's going to require some tools like this, but with some careful planning and thinking, you're going to find some students that are going to be absolutely blown away and intrigued by the science here.
Okay.
I think I should stop.
Yep, I need to steal this away, and thank you for our session, that great session. We will stay on as long as we're allowed and answer any questions. If you signed up for a free student modeling pack, we will send you an email about the CRISPR webinar series. Okay, so you'll be able to get more information about that. And we'll also send you information about our summer courses. We do a whole bunch of different summer courses, from modeling the molecular world to 3D printing. So in addition to this genome editing. In the session today, Tim used a bunch of different materials. So we have the nucleotide student modeling pack, our adaptive immunity kit, the making the cut, and we also have a mini model version of the 3D printed model that Tim had available. You can purchase any of these at 3dmoleculardesigns.com, but use the CASE21 discount code to get 20% off most of our products, okay? And if you would like to borrow models, you can borrow them from the lending library, and hopefully the CRISPR materials will be in there soon. I'm realizing they may or may not be.
That's a good point.
CDM will be adding them soon.
Yep.
So thank you. And Jennifer, did you have anything that you wanted to add for the closing here?
No, thank you. That was really informative. I appreciate seeing this model. It's really cool. And if you want, I did mention the feedback form that is actually back on the platform of the conference. So when you go back to your, you registered or like put this in your schedule, there should be a survey after this. And I did put in the chat that the recording will be available at the end of the month, but if you want it earlier than that, to contact CASE for that. So thank you. Great session.
Can I just say one last thing? If you like this model, you should go on this thing called Google and Google Jennifer Doudna. And if you search through all the images of Jennifer Doudna, you will find several in which she is holding her model of Cas9. So this model was actually made with Jennifer and her group as they were opening their new Innovative Genomics Institute at UC Berkeley. So she even talks in her book about how she took this model into her son or her daughter, one of her kids' middle school classrooms and explained CRISPR to them using this model. So it's a good story.
Great, and we are now at 4:15. So if you have any questions, stay on and ask them. If not, I hope that you can join us for one of our other sessions. So today at 4:30, Chris Chow is doing Exploring the Wonderful World of Cell Membranes. And then tomorrow we have a using models to make connections between DNA and chromosomes. So I hope that you could join us for one of those. So thank you, everyone.
Okay.
Thank you so